sorry, I know that's startling to people. Oh. Awesome. Yeah, first hour we covered mainly aerobic respiration, Ryan. So we went through all of those processes that Tyler listed. And I am recording, so these will be posted on the Moodle page, hopefully by 1 today at the latest. It'll depend on how long the recordings take the process. But we're back at it. So our last hour, I have kind of a little bit of popcorn of things I want us to cover. I want to talk about ketogenesis, as well as ketone degradation. We'll also probably spend a moment on the pentose phosphate pathway, as well as the urea cycle. So those are definitely things I want to cover. There's many more things in this packet that you can cover, but these are the ones that are the most high yield at this point in your study, about a week and a half away from your test date. So a reminder about ketone body metabolism. We can see here that in states where we're having a lot of lipolysis, we are going to have a large amount of free fatty acids then in the plasma, because we've broken down those fatty acids into their free fatty acid form. We will then see an increased amount of hepatic output of ketone bodies, and we will get into what's called ketoacidosis. So our ketone body metabolism, we can see here acetoacetate and beta hydroxybutyrate are two of the culprits that we will most commonly see as ketone bodies. And if you look at who likes to use or who can use these tissues during a starvation or fasting state, the nervous system and the brain, remember, they primarily are using glucose because glucose is their main form of fuel, but they can use ketone bodies if needed. They are not able to use fatty acids. The rest of the tissues, skeletal, heart, liver, kidney, can use fatty acids in a starvation state, and they will. They will let all the glucose go to the nervous system. So any glucose that's left when you're in a starvation fasting state will go to the nervous system. And then the intestinal epithelial cells are interesting in that they don't use glucose or fatty acids, but only ketone bodies in a fasting state. Liver will not use ketone bodies in a fasting state, so liver and brain are similar in that process. So kind of keeping those pieces in mind, those can be high yield questions because they're connecting you back to physiology and to anatomy. So I want to talk about actually how we get here. This process here, we can see here our liver process over in this section. This is peripheral tissue / blood. And then this is actually like peripheral tissue. So for example, muscle or the heart or etc. I'm going to go back to my main PowerPoint. This is the one we started with, the biochemistry PowerPoint. We were on this before in the first hour, and we're going to go into ketogenesis, the generation of ketones. So this process occurs in our liver primarily. It occurs within the mitochondrial matrix. Our substrate, again, is acetyl-CoA. So again, super important piece of the puzzle. A key step is HMG-CoA synthase. We also can see, HMG-CoA synthase is going to be your key enzymatic step. So not to be confused with HMG-CoA reductase, right? HMG-CoA reductase in cholesterol synthesis, HMG-CoA synthase in ketone synthesis or ketogenesis. Your products are going to be beta hydroxybutyrate acetone and acetoacetate. So those are going to be your three products. Ignore the dehydrogenase. That's an enzyme. Beta hydroxybutyrate, acetone, and acetoacetate. It's regulated by your concentration of your substrate, so acetyl-CoA, and your key intermediate is HMG-CoA. It is going to require a little bit of energy input, so 2 acetyl-CoAs and 1 NADH. So we can see here in this process, we have acetyl-CoA going to acetyl-CoA, HMG-CoA, and then we have the actual initial ketone, acetoacetate, which can be converted into acetone, which will often expel. You can see it in a breath. It's very sweet smelling, or beta hydroxybutyrate. And these ketones can then move into circulation and be taken up in places like the brain. Beta hydroxybutyrate then can be converted into acetoacetate, and it can be used to get us back to acetyl-CoA to form energy. Looking at it from a different perspective, back to our PDF, we can see this process in less of kind of a pretty form. So ketone body synthesis in the liver of the mitochondria. We start with our acetyl-CoA, 2 of them. We are going to then combine them together to get acetyl-CoA. And then we have our rate limiting enzyme step, HMG-CoA synthase. It's going to essentially take off that CoAsh portion. So that sulfur component is going to get removed. And so we go from acetyl-CoA to our intermediary step, 3-hydroxy-3-methyl-glutarol, or HMG-CoA. This is that intermediary step. So this would be our rate limiting step. If this doesn't go forward, then we're not going to create ketones. From here, we are going to essentially break off the acetyl-CoA portion of this HMG product, and that gets us acetoacetate, the first ketone formed. Acetoacetate can spontaneously generate acetone, or it can be converted to beta-hydroxybutyrate via beta-hydroxybutyrate dehydrogenase. And in that process, that's when we use that energy molecule. So that's formation of our ketones. The breakdown of our ketones, we can see it's going in just a different direction. So ketone body breakdown in brain, skeletal muscle, kidney, gut, not liver. We can see beta-hydroxybutyrate is going to get converted back into acetoacetate using, again, beta-hydroxybutyrate dehydrogenase. This can go either direction. We then gain an NADH in this process. Acetoacetate now does not go back in the same process here. It has a different enzyme, succinyl-CoA, acetoacetate transferase. This is a different enzyme used. We then have acetoacetyl-CoA, and then thiolase, again, same enzyme at the beginning, can go either direction to get to acetoacetyl to acetyl-CoA. So our rate limiting enzyme of this step is this enzyme here, succinyl-CoA, or acetoacetate-CoA transferase. This is our rate limiting step for ketone body breakdown. Okay. If you wanted a different version of it, you can see here brain, skeletal muscle, heart within mitochondrial matrix. Breaking things down, I have beta-hydroxybutyrate dehydrogenase here as a key step, but it's not the rate limiting step on this. It's what I mentioned before. Product is acetyl-CoA, HMG-CoA is your intermediate, and then this is your energy production. 2 acetyl-CoA, 1 NADH, and you do have to use a GTP. Okay. The most common ketone questions I see are asking about the 3 types of ketones, or the 3 actual products that you can have in the body: acetoacetate, beta-hydroxybutyrate, and acetone. Beta-hydroxybutyrate is typically the one that is going to move through circulation. So beta-hydroxybutyrate gets acetoacetate can as well, and can get taken up into the brain, into the tissues. But beta-hydroxybutyrate is often the one that we see transferred around the tissues, taken up and utilized the most commonly. Acetoacetate would be your second most common, and then acetone is not. Acetone is exhaled as a byproduct, typically. Okay. Great. All right. Kind of 2 more funky pathways, and then if we have time, we'll look at some vitamin stuff and minerals. This is the pentose phosphate pathway. There's only a few things I want to say about this. Essentially, you also could call this the HMP shunt. It helps create NADPH. That's its product. It also gives a byproduct of ribose sugars. NADPH you need for any of your reductive reactions. So glutathione reduction inside red blood cells, fatty acid and cholesterol biosynthesis. So it's a helpful product to have. And so this is part of how we can generate NADPH in the body. It also gives ribose, which can be used for nucleotide synthesis. So that is also an important thing, because we need nucleotides as our backbones for our DNA. There's 2 main phases here: oxidative, which is represented in blue, and non-oxidative, which is represented in green. Both of these occur in the cytoplasm. So we're not having this take place in mitochondria. So it's different in that sense. And there's no ATP used, and there's no ATP produced. We can see this process occurring in the liver, mammary glands, red blood cells, adrenal cortex. Those are the main sites that this happens, typically sites of fatty acid or steroid synthesis. But again, red blood cells, that's an interesting one, because that's red blood cells we don't think as much as having a lot of activity intracellularly, because they're anuclear, but they actually do a lot. Your substrate that you're using is typically glucose 6-phosphate. So we can see glucose here getting converted via hexokinase into glucose 6-phosphate. Then our key enzymatic step would be glucose 6-phosphate dehydrogenase. That goes from glucose 6-phosphate into this product here, which is phosphogluconate or 6-phosphogluconate. So taking this here and pushing it over into this oxidative pathway, because this is how we get it to commit essentially to the pentose phosphate pathway. That's why glucose 6-phosphate dehydrogenase, the enzyme that governs this conversion, this oxidative step, is our main key enzymatic step, our main rate limiting enzyme. There's also another one that is important, transketolase. Transketolase comes much later, taking us to ribose 5-phosphate. Where is that? Where's my ribose? Ribose 5-phosphate transketolase. So taking us essentially multiple different ways. You can see it's kind of going all over the place here, but transketolase can take ribose 5-phosphate transketolase and can go back to fructose 6-phosphate. So it can go from here all the way up through here. They're kind of like all these crazy steps. It can also take ribose 5-phosphate and create our glyceraldehyde 3-phosphate or GAP, which can then also kind of go back in that glycolysis cycle. So transketolase is important because it can kind of connect things back into this normal kind of glycolysis pathway if needed. If we need to move back down here, if that's more important than generating NADPH or using actually the ribose 5-phosphate for something else. But if you had to choose one enzyme, it'd be that first one, glucose 6-P dehydrogenase. NADPH in ribose is your big products. That's really all I would want you to care about for the pentose phosphate pathway. Know where it's at, cytosol, substrate, product, and key enzymatic step.
Dr. Petro.
Yes.
So I read a lot about like G6PD deficiency.
Yes.
And I was just wondering, like, obviously that has to do with this pathway, but
yes,
I didn't really understand why or like how. I saw that it causes hemolytic anemia, and I kind of like, it kind of lost me there.
Totally. So that's a great piece. So G6PD deficiency, so glucose 6-phosphate dehydrogenase deficiency is what you're referring to here. And so NADPH, which you get as a byproduct from this pathway, is needed to keep glutathione reduced in its reduced state. When glutathione is in its reduced state, it will help with detoxification of free radicals as well as detoxification of peroxides in the body. When you have a decrease in NADPH in its reduced state, especially in red blood cells, which this process occurs in red blood cells itself, that can lead to hemolytic anemia because you don't have adequate red blood cell defense against oxidizing agents. So typically, if you have an adequate store of NADPH, you're going to have nice amounts of glutathione present so that if free radicals are in the body, which they are constantly from things we consume to things that we're exposed to, the body can protect those red blood cells via NADPH and glutathione. When NADPH isn't present, because we weren't able to convert glucose 6-phosphate into this pathway, we weren't able to shunt it over because this enzyme that goes from here to here was lacking, then we don't have that glutathione present. We aren't able to defend our red blood cells against these oxidizing agents. And the oxidizing agents, what they'll do to RBCs is they're going to essentially cause hemolytic cell death, because either through apoptosis, programmed cell death, they're damaging to red blood cells. And that's part of, if you think about an oxidative species, anything that's oxidative can be damaging to tissues if we don't know how to deoxidize it. So if we look here, I'll show you this. I think this will help you visualize glutathione. There we go. There we go. Okay. So glutathione in kind of oxidant generation. So in this process here, we have glucose 6-phosphate, these two different pathways. We can see glucose 6-phosphate going into glycolysis, glucose 6-phosphate going through essentially kind of this pentose phosphate pathway. So glucose 6-phosphate, if we have glucose 6-phosphate dehydrogenase, we can generate NADPH. as well as phosphogluconate through the pentose phosphate pathway. When we have NADPH, we can, through glutathione reductase, we can kind of keep this cycle going. This keeps the red blood cells healthy by being able, if we have oxidative stressors, like from infection, drugs, even fava beans, so things like our OH or H2O2, any reactive oxidative species, those can get converted into water using glutathione peroxidase. If we have glutathione reductase, we only have these able to turn the story if we have adequate NADPH. So if we stop this process here, we can't turn this, meaning this won't turn, meaning this won't turn, meaning this won't turn, meaning that we'll keep these reactive oxidative species building up within our red blood cell, which will cause the red blood cell to lyse.
Thank
Did
you.
that answer it?
Yeah, that was awesome. Thank you.
Okay, yeah, absolutely. So would glutathione injections be beneficial with patients with G6PD deficiency? Well, so you could see if we add just glutathione here, this is glutathione reductase. So could glutathione be helpful? Sure, but if we still don't have this enzyme present, we can't turn this cycle irregardless, right? So even if we had the glutathione here, we really need glutathione reductase to be able to actually do its job of converting NADPH to NAD+. It's not for necessarily a lack of glutathione concentration, it's a lack of this enzyme. Glutathione injections could help your body's ability to process if you have like a partial deletion of this enzyme. So if you had a partial deficiency, maybe not a full deletion of this enzyme, but maybe it just wasn't working as well, then yes, glutathione injection could be helpful to up this concentration to make sure that it's able to do the maximum amount of the job for however much you have. But if you have a true deficiency where you're not creating any of this enzyme, your treatment you would want to make sure you are actually promoting or repleting that deficiency. Did that answer it, Gigi? Awesome, thanks.
The other piece, I'm glad that you went to this picture, because I was going to ask the glutathione connection too, because that's what I remembered. And then the one other thing that's also on there is that the Heinz bodies end up precipitating out. And so as I understand it, you can see these little precipitates, and then sometimes also the macrophages in the spleen will like take bites out of the blood cells. Which is called
bite cells. Yes.
Yeah.
Yep.
Yeah.
So
those are the connections I remembered from the HMP shunt also. Yeah.
That's perfect. Yeah, your Heinz bodies, like Heinz ketchup. So Heinz bodies are just denatured, broken down globulin protein chains. So essentially precipitates that you can see in the red blood cells due to this oxidative stress or damage. And then phagocytes are going to try to remove them. So you'll see then bite cells from phagocytic removal of the Heinz bodies by spleen macrophages, right? Because the spleen, we're trying to clean the blood. So if the macrophages in the spleen notice that there's some kind of damaged blood cells with these Heinz bodies, they will bite into them to actually clean up on IL-2, clean up the spilled Heinz ketchup. So those macrophages are gonna bite into some Heinz ketchup. So you could remember your bite cells with your Heinz bodies.
Yeah. It's just crazy to me that the body can actually, like, it won't kill the whole cell. It'll just try to
bite it out. If it can. If it can, it tries, but often it will end up as cell lysing. You'll often lead to hemolysis. That's great. And that's a great connection. If you're like having trouble remembering your free radical generations too, on this page, this kind of talks about oxygen to superoxide, hydrogen peroxide, and then your hydroxyl radical. So kind of like this process here from oxygen back to water, going through your free radical generations. And this can be from a whole host of things that can cause damage, infection, different microbes, lots of things can kind of lead to having these reactive oxygen species present in the body. Cool. This was actually a great time, and I'm glad we went here next because that's where our pentose pathway is, so it makes sense. So again, pentose phosphate pathway, then we got into our oxidative reactions, and that led us down to oxidant degeneration or oxidant generation. Another kind of component, if you are wanting to also look at where NADPH can be used, you can see it used in these respiratory burst, essentially cells or respiratory burst reactions. This is another way that H oxidase can be used. And if you have a deficiency in this enzyme, you can also get buildup of granulomas or buildup of these reactive oxygen species. I would say this scary process is much less important to understand than this process of glutathione. This is much more commonly asked about. I've never seen respiratory burst asked about, except for in the context of understanding this conversion of oxygen to superoxide, to hydrogen peroxide, to hydroxyl radical. So if you understand kind of this process here, stepwise, that plus this would probably be what you'd be asked about regarding reactive oxygen species. Okay, great. Let's see here. That lets us into our fat. Okay, I had another place I was going to have us go. Oh yeah, one other cycle, and then we can look at vitamins. So the last cycle I wanted to go into is the urea cycle, because again, this is part of our waste cycle in the body. So the urea cycle is going to occur in the mitochondria as well as cytosol of liver cells. So you can see there's components that occur in the cytosol and components that occur in the mitochondria. And really our goal with the urea cycle is to help with our elimination of urea into our urine. And so that's kind of part of what we're trying to do. To create urea, this process, we are also pulling in other byproducts that we want to get rid of in the body. Urea, the makeup is going to be 1 NH2 from ammonia, NH4. So we're getting rid of that toxic compound. A carbon and oxygen, so carbon double bound to oxygen, so CO2, and then an NH2 from aspartate. So these are kind of the makeup of how urea is built. It takes a couple different components. The most common cycle defect we'll see is ornithine transcarbamylase enzyme. That's here. So the second step is where we see the most common defect in people or deficiency. But we'll just walk through this pathway together. So inside your mitochondria, you have carbon dioxide and water. They're creating bicarb. Bicarb plus ammonia, NH4, will be combined together in this first step, which uses 2 molecules of energy, 2 ATPs. This is the high energy use step using carbamoyl phosphate synthetase 1. From this step, you have your carbamoyl phosphate. You are then going to combine that with ornithine, which ornithine you'll see is actually in the urea cycle. So this is a urea cycle. This is a cycle that's going to go round and round and round. This ornithine does have to get shunted inside the mitochondria and to be able to be used, which is why you'll have it shunted in. It gets converted into citrulline, citrulline shunted out. So this is an ornithine citrulline exchanger. Ornithine goes in, citrulline comes out, and this process can continue. That's how this isn't fully depleted, because these are essentially exchanging one for the other. This is the step from ornithine to citrulline that most often is deficient or defunct in this pathway. So this would be an important enzyme to know. Citrulline then gets shunted back out. You add aspartate. Citrulline plus aspartate gives you arginosuccinate using an enzyme of that name. Arginosuccinate then will become arginine and fumarate. Fumarate is off as a byproduct via a lyase, breaking a bond. And then arginine will become ornithine, and the byproduct here is urea, which can then go into the urine. If this process doesn't work, if this enzyme isn't working, we can have buildup of these back products, which could be carbamoyl phosphate, which then would lead to a buildup of bicarb and ammonia or ammonium, which would be toxic to the body. We can see a buildup of ammonium for multiple different reasons, multiple different ways. We often see it with liver damage, cirrhosis, or fibrosis, because again, this is occurring in liver cells and hepatocytes. So if the liver has damage or death or dysfunction, and this process can't go through, that's how you can get ammonia or ammonium buildup. And that's why you can potentially see liver damage leading to high levels of ammonia, leading to potential cerebral changes or delirium changes that are temporary until you fix this ammonium level and essentially improve that conversion pathway again. Any questions about the urea cycle? Key pieces: using ammonia and bicarb, ornithine transcarbamylase, happening in both mitochondria and cytosol of liver cells, urea as your product going to urine. Okay,
Does it just
Yeah.
occur in the liver?
correct.
So how does but like the toxic, what is it? Ammonia is the toxic element that gets transported to the liver for
Correct.
removal?
Yes, for removal.
Yeah.
You can have buildup in the blood, and then the body, the liver cells will take it up. And you also have breakdown of it in liver cells as well, because this is a breakdown of what? Well, how do we get ammonia? Breakdown of proteins, right? Breakdown of amino acids. The breakdown of amino acids is occurring in the liver.
Mhm.
So that's how you're getting this product specifically in the liver. Does that make more sense?
Mhm, mhm.
So you'll see amino acid degradation and protein degradation, which can lead to that byproduct.
Mhm.
I don't have it here. So here we go. I'm not going to go through all of amino acids and proteins. This is like all the branched chain amino acid breakdown, amino acid derivatives. I don't find them to test that much on this stuff up here, but I do see them often ask some amino acid for neurotransmitter questions. So which ones would I have us focus on? Glutamate to GABA can be good. I would really, honestly, I wouldn't worry as much about like this process as much as the amino acid derivative for these products. So histidine going to histamine. That I would put that to your memory. I wouldn't worry as much about actually knowing the exact process at this point. You know, glutamate to GABA. I think like that's high yield. Methionine to CM. That's your S-adenosylmethionine essentially. Those pieces, glycine and arginine to creatinine. I think those components, like knowing those pathways, tryptophan to serotonin, melatonin, that association, great. The pathway to get there, probably not high yield for you right now. Okay. Phenylalanine, dopamine, epinephrine, norepinephrine, like knowing that's your pathway, great, because phenylalanine goes to tyrosine, dopamine. But I don't believe you actually really need to know each kind of step. So this is your summary slide. So I would focus on this as your high yield page. It talks about your amino acid derivative and then the products it can go to, and then some of the important cofactors that you might need. So B6 you can see is used in a lot of them, even here, and you can also see vitamin C here. And then some of these byproducts like tyrosine to thyroxine, our thyroid hormone connection, dopa to melanin, our skin pigmentation connection, et cetera, et cetera. So this is what I would focus on here for this kind of component versus the actual metabolic biochemical pathway. When it comes to heme synthesis, really understanding that like the heme molecule and that there's an iron in the center and that's what's really binding your oxygen or your carbon dioxide. I think that is more ideal to understand right now, high yield wise. Heme degradation, I would say same. Phase one, go ahead.
On the phenylalanine, the catecholamine pathway.
Yeah.
I thought I was always read that was tyrosine, but so is it important to know phenylalanine instead for
So
some reason?
phenylalanine is first, right? And then that goes to tyrosine. So you're right. Tyrosine is here as that backbone, but phenylalanine is what actually is going to get converted into tyrosine. Tyrosine then gets converted into the rest of them. So you're correct, and this is correct, right? So tyrosine, great, but they've also asked, what's the backbone of that? Phenylalanine. And so they've said, what's the precursor for dopamine, and given you like phenylalanine, tryptophan, arginine, and glutamate, and tyrosine's not an option. And so phenylalanine would be your choice.
Oh, see, that's weird because I only remember tyrosine, but okay.
Because all we ever teach is tyrosine.
Yeah.
Yep, that's all we ever teach. But the actual like true precursor, which I don't know, would be phenylalanine. Okay. Yeah. That's all it's ever talked about. We talked about supplementing with tyrosine or, anyways, yeah. Good question. Just to clarify, 32 ATP from 1 glucose, 23 from ketone degradation. I can confirm 32 from 1 glucose. I'd need to look at ketone degradation to confirm 23, but it does sound right to me, much less than glucose, but still decent.
Is there some weird 30 pathway also,
Yes.
like
So
in
in
a
the
different
first
cell type or something?
in the first hour, we talked about the 2 types of shuttles. So I'd encourage you to watch that. It's towards the end of the lecture, but I'll pull it up here.
Does that matter ever? Like,
They
is that
could
just like
could ask you, I mean, I don't know. So the malate shuttle, really what they would ask
Yeah.
is, so the malate shuttle occurs in the kidney, liver, heart. This will give you your 32 ATP molecules. These are in areas that you have lots of NADH, so you don't minus any. The glycerol 3-phosphate shuttle, this is in brain and skeletal muscle. This is when you actually expending 2 ATP in the form of these NADH, FADHs. So you end up with 30 ATP total. So in brain and skeletal muscle, you'll generate a little less ATP for 1 molecule of glucose than you will in kidney, liver, and heart.
Yeah. Okay. Thanks.
No, you're doing great. You're awesome. Okay. Let me go back down over to heme. I'm going to go back down to the bottom so we can get into our last few pieces. While I'm going here, I'm just going to go through some high yield vitamin stuff, but if anyone has questions on things we've covered so far or other areas of biochemistry, I'm happy to also kind of popcorn out questions for the last 30 minutes as I kind of go through some high yield stuff. All right. Well, pop in if you have something for me. Okay. So the vitamins here, this just breaks them into, this is page 92. So this just breaks them down into water and fat soluble, which you probably already know. Non-B complex water solubles and B complex water solubles. And then this one has kind of talked about things that can be more energy releasing, involved in hematopoiesis, so folic acid or B12, or have other properties. I don't know how much helpful that is. It's a table, so there you go. This pathway, or this table, it's really hard to see. I think the only helpful thing, because it's like super challenging to see here, is the names. Understanding like vitamin B12 has these other names that can occur in the body, and ascorbic acid. They won't always say like B1, they'll say thiamine, right? So understanding that these can be used, but they mean like thiamine pyrophosphate is literally like vitamin B1, but in its active form. Specifically in those 2 big enzymes we talked about at the beginning, alpha ketoglutarate and pyruvate dehydrogenase. So we can see here like where they could be biochemically utilized. That's the active form versus the other names for kind of their common vitamin name. Same for our fat soluble vitamins. And so you often will see like ergocalciferol or cholecalciferol instead of vitamin D on these tests. So that's where this table could be helpful just for like that name recognition. Looking at folate a little bit closer, when typically folate comes up on the exam is talking about birth defects or folate deficiency. We can see this folate structure here. It does contain a glutamate in it. It is going to require NADPH to be converted from folate to dihydrofolate, and then it's going to need another NADPH to be converted from dihydrofolate to tetrahydrofolate or FH4. From there, we can see FH4 getting broken down into more and more steps. And then bingo, we see the MHTFR conversion right here from this methylene FH4 to N5 methyl FH4. So this is the step that if you have an MHTFR defect, whether it's partial or complete, it can affect this folate pathway in the body. And this is where we could potentially see some birth defects or even just adult issues with B vitamin processing, especially if it's not already methylated when we take it in. This is connecting you kind of back to sources of carbon for the FH4 pool and recipients of the carbons. This is kind of showing you the backbone of like, these are different amino acids that play with folate in this process. Okay. But I think this would probably be the best connection, is just to know you're utilizing NADPH, so you still need that pentose phosphate pathway, that cycle. You're utilizing some energy in the form of ADP and NADH, and this is where the MTHFR gene comes into play.
Would a neural tube defect be the abnormalities you're talking about?
You could have a neural tube defect, yes, especially if you had like a complete defect, but typically you see neural tube defects when not necessarily when you don't have this appropriate gene, but when you don't have appropriate starting concentration. So that's when you see the most neural tube defects from folate, is when you don't actually have the concentration present, which is why we recommend mothers who are interested or people who are interested in achieving pregnancy to supplement with folate prior to pregnancy. And then obviously checking your folate level if you're not absorbing it or actually translating it into its active form. Say, do you potentially have this defect? If so, then we would want to make sure we're already giving you a methylated form, so that you don't have to go through this conversion factor to methylate it yourself. So MTHFR gene mutations, the two that are most studied are C677T and A1298C. Do I ever memorize those? Never. Just recognize, so we can see kind of frequencies and things like this, percent of normal enzyme activity. Really recognize that if there's no variance, you have 100% effect of your enzyme. But if you have a lot of variance, you're going to have less effect of your enzyme. So it's a spectrum. Some people will have like just a mild decrease in effectiveness of their enzyme, their MTHFR. Some people will have a great defect, and so they'll have a greatly decreased activity, 24%, instead of someone who had a normal no variant MTHFR gene. So there's a scale, and that's why some patients with MTHFR have really no symptoms or low symptoms, because they're still able to do the pathway most of the time. And then some patients will be debilitated or have really significant symptoms from this gene defect. B12 here, we are seeing B12, it'll be synthesized by bacteria, it's found in animal products, and it's stored in the liver. So we take up, when we eat B12 products, containing products in our diet, and parietal cells from our gut secrete intrinsic factor, we're able to bind these two together. It creates a glycoprotein, a sugar protein, and we're able to actually absorb this in the ileum. We're able to take it through our mucosal cells. Essentially, intrinsic factor is the binding agent. B12 then is able to be absorbed and then shunted into the blood where it's bound to a binding protein. If you don't have intrinsic factor, you will not be able to absorb B12. And so that's how you can get those pernicious anemias where you're unable to actually, you don't have intrinsic factor, which leads to that B12 deficiency. Or if you do have B12, but maybe, or you do have intrinsic factor, but maybe you're not intaking animal products, or maybe you have a partial defect, but not a full loss, you could see your B12 deficiency, your megaloblastic macrocytic anemia, like we discussed yesterday. All right, that's my big thing with B12. With vitamin D, we have sunlight that's going to start this process from 7-dehydrocholesterol here, yay sun, to cholecalciferol here. That occurs in our skin. That's then going to be absorbed, transported via circulation, portal circulation to the liver, where it's then converted into its 25-hydroxy D form or 25-hydrocholecalciferol, which then will be moved from the liver to the kidney, where then we can convert it into our 1,20-dehydrocholecalciferol, which is essentially our this is like the active form to our actual usable form in the body. But we see there's 3 different organs or 3 different sites that vitamin D conversion takes. What does that mean? A lot of ways for our body to not do it well, right? We have to have healthy skin. We have to have healthy liver. We need to have healthy kidneys to do this pathway appropriately. Low amounts of phosphate and circulating parathyroid hormone is going to increase the process from 25-hydroxy to 1,25-dehydroxy. This is going to also exert some negative feedback. When we measure vitamin D, we measure this structure or substance here, because this is the one that's going to go through portal circulation to get to the kidney. So we are able to measure this in the blood. You also can measure this kidney byproduct, but typically we are not measuring this unless if we're concerned about actual kidney disease. When we're worried about vitamin D levels, like are we absorbing, are we actually having adequate levels? Typically you're measuring the 25-hydroxy form, and that's what we use to decide if we're going to supplement or not. if you're concerned about a kidney disease or dysfunction affecting your conversion here, you then would actually measure this 125 dehydroxy form. And I have used this sometimes in patients that I know have a chronic kidney disease, and we'll measure this value. This is the value that will then act on our cells to then increase calcium absorption across our intestinal lumen, across our intestinal cells, to actually help support calcium concentration in the blood and in the bone. That's that piece there. So vitamin D relates to 3 organs: sunlight, phosphorus, calcium, parathyroid, and calcitonin. We have all those pieces of the puzzle that are involved in our vitamin D metabolism.
And then you can also get it through your diet, right?
In
A
very
different
small
form.
amounts, but yes,
Um,
you can.
Do you think there's I guess this is a rabbit hole, but is there a difference between dietary and UV produced? I've heard there is, but I don't know if that's just.
There is. I forget the form that you absorb it in your gut. I do know it's less absorbable, so you have a harder time actually absorbing it through the intestinal lumen, and then you have a harder time converting it into its active form if it comes from the digestive system. So I need to go down the rabbit hole to figure out what form you would consume in food. Fish has a higher concentration than most foods, but I do know that it tends to not be absorbed as well and tends to not be able to be converted into the active form as well if you get it through your
I think
digestive
that
tract.
was what I was hearing.
That's
It's just
probably it.
interesting because we often say like, take dietary vitamin D, and it's like,
Yep.
well, maybe we should look towards like UV, a little UV exposure to get more
Well, sunshine
effect.
in your life. Yeah. So vitamin D3, this form is typically what we try to supplement with. But you'll often, like the pharmaceutical vitamin D that's covered is vitamin D2, which is cholecalciferol. And that one is more challenging to absorb into the diet, into the gut. So that's the dietary form. I don't know if, or vitamin D2, vitamin D3. I don't know what form it comes in food. So that would be the one thing. But supplementation wise, it's either D3 or D2 that you're getting. Yeah. Excellent. Vitamin A, rare that they test you on vitamin A in like this synthesis process. Just really the piece that I've seen is that your dietary source is beta carotene for the most part, and then that gets converted into retinol and retinol with an AL and retinol with an OL, and then an ester from there, and then you transfer it via chylomicron because it's a fat, right? That would be the biggest one, is just remembering beta carotene, your dietary source. Vitamin K more in your clotting cascade. We're going to look at that, I believe, when we talk about clotting pathologies. Biotin, I don't think that's as high yield. And then this page here kind of takes you through what our carbohydrate metabolism discussion was and kind of puts in different vitamin cofactors in places. You can see like our 3 vitamins here with our pyruvate for pyruvate dehydrogenase. Do I think this is high yield at this point? I don't know. You're close to your test date. I wouldn't worry about it. Here's a summary table. This is coming from USMLE. Another summary table showing kind of where things are doing things. And then your vitamin summary table at the end. If you just really want to get lost in the sauce and look through all this, go for it. We'll do in genetics tomorrow, so we're not gonna look at that yet. Okay.
I have a quick clotting cascade question.
Please, I love clotting cascade questions.
I think I do too, but the one thing that I'm stuck on is like the initiation of both. Is it that trauma, like any type of trauma initiates both cascades, or is it that like, does like an actual cut only initiate one? Like that's kind of where I get a little lost.
Yeah, so that's great. So exogenous versus endogenous clotting cascades. So just think about where the source of the trauma is. If it's endogenous, if it's something that's happening within the body, blood vessels damage internally, that can start the endogenous pathway, versus an external process would be something happened to you, like a stab wound, a cut, could be a microbe, that could potentially start the exogenous pathway. Does that help or make it worse? I don't know where you went.
Oh, sorry, I was muted. So the intrinsic pathway would be the endogenous pathway.
Endogenous pathway, correct.
Okay, awesome.
Extrinsic would be exogenous. Yeah.
Great. Thank you.
Yeah, absolutely. Can they both get activated? Sure. Excellent. What other questions do you have? Biochemistry. Good, the bad, the ugly. Always good to know your amino acids. Your essential amino acids, that could be a helpful one. There's always going to be more to know for biochemistry, so don't worry about knowing it all.
I got one that's, well, it might be too much, but like I know, you know, the amines and this presence of nitrogen, like, do you have any sort of thoughts on like what is it about nitrogen that is needed, and like why is it, but it's also toxic, so it's just this biochem puzzle that I've been trying to figure out. Like, sort of like, you know, we're carbon-based life because we form these long chains because of the carbon atom. Is there something about the nitrogen, the properties of nitrogen, that make it suited to, like, we just have to have it?
Yes, good question. So our properties of nitrogen, remember, nitrogen is typically bound to 2, 3, or 4 hydrogens, depending on the form. All of our amino groups really come from our amino acids, which are the building blocks of our proteins. And to actually create those peptide bonds, we need the amino group to react with the carboxylic acid group. So that NH3 to react with the COO. If that doesn't take place, if we didn't have the amine groups, we wouldn't have the actual tight bond that's needed for a peptide to then be able to make our secondary, tertiary, and quaternary conformations of proteins. So part of the key picture of having nitrogen in the body is really the formation of the peptide bond, which is between an amine group and a carboxylic acid group. Without that amine carboxylic acid relationship, the bond would not be as strong, because carbon-carbon bonds are strong, but this bond is, the peptide bond is particularly strong because there also is that charge gradient as well. And it sets up the position for our body to be able to then create either the kink, the alpha conformation, or the helical conformation, alpha helix or beta plated sheet, right? To be able to then set up our proteins to fold. So that's a big, that I would say is probably the crux of why amine groups are in the body, even though, and that's why the breakdown of proteins and amino acids leads to that ammonia buildup, that toxicity.
Yeah. Okay, thanks. Yeah, that helps me out.
Yeah. I saw your hand, Claire.
Yeah, so I know we talked about this one-on-one, but at this point, a week and a half away, if you're not a big biochem person, what should we really commit?
Commit to that table, memorize or study that table, practice filling that out. That's what I would do.
Okay, thanks.
Yep. And the table, it's posted on there. It's the biochem pathways blank, and then there's one that's partially filled out, and then there's one that's fully filled out. It's like an answer sheet. So that's what I would commit to a week and a half out. If you already have that down and you're looking for a secondary step, then I would start leaning into some of these like things that involve vitamins and minerals and cofactors. That tends to be the next more high yield. Great. What other questions do we have? Oh, as I was like flipping through my little notes for y'all, I came up with our essential amino acids. If you don't remember the most commonly associated acronym, it's private Tim Hall. These are your essential amino acids: phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, leucine, lysine. These are the amino acids you have to obtain from diet to live.
What is it one more time?
Private Tim Hall. So phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, leucine, and lysine. The A in Hall does not represent anything.
Great.
Yep. Awesome. Another one that you could be asked, let me pull up here my little notes. my handy dandy notebook. Sorry, I don't bust this out that often until I do this class. I'll pull it up. Is your your essential fatty acids. So remember, your omega-3 fatty acid is your linolenic. That's going to be EPA and DHA. So I'll write this: omega-3, linolenic, EPA, and DHA. Remember that your omega-6 is linoleic, which can also go into your arachidonic acid. Oh, whoops, that converted itself. Linoleic. So these are things that you would need to consume via diet. And obviously, we're typically heavier on the omega-6 than the omega-3, which is why omega-3 supplementation has come into claim to fame. Anything else? Any other questions that you can think of? Questions, concerns?
Is the plan to meet in person tomorrow?
Yeah, unless I'm 5, it's screwed again, but it shouldn't be.
Okay,
Yeah,
great.
that's my hope. Absolutely. I was on the 5 from 6:30 to 8 a.m. guys, before I was able to turn around and get home. So I live up in Washington, so I don't know if anyone else got stuck in that, but yes, we will meet tomorrow. 8 a.m. will be our start time as usual. The first 2 hours we are going to cover genetics. The first hour, it's going to be generic genetic mechanisms, and what I mean by that is inheritance patterns. So sex-linked, recessive, dominance, codominance, as well as some basic genetic replication components, just to remind ourselves. Within that, I'll kind of include some of the potential mutations and also conditions that would go well within that discussion. Our second hour will be more the genetic SNPs, the SNPs, the specific things that are more high yield, that list. And then the last 2 hours, we'll start going into our pathology discussion. Our path discussion, we'll start with more of the higher yield, larger systems, and then we'll move down from there. I think we have a whole day for path next week as well. Okay, awesome. Send me questions. Oh, go ahead. Yeah.
Sorry, just while you're on path, with the biochem path stuff, there's a lot of like glycogen storage and like enzyme deficiencies. Do you have any like high yield recommendations for where to focus for those?
Yeah,
because I get real confused with those.
Yeah, I would say our G6PD deficiency would probably be your most high yield, just because it's commonly discussed in naturopathic circles, or we're testing it for IV therapy, so it's more high yield than a lot of them. We'll discuss a few of them in our path lecture. I would lean more into ones that are associated with, I would honestly focus on G6PD. I wouldn't spend too much time necessarily going down the weeds. G6PD or your enzyme deficiencies associated with steroid synthesis. That would probably be the areas that you would be tested on the most. They would be the most high yield at this point.
Okay, thank you.
Yeah, of course. There's so many that you could go through. I just don't think it's worth a lot of your time. You'll get a few questions on those. Great.
I was also wondering, for what do you think about like spinal cord, like the different like lesions at the different levels of it? I feel like I got so confused during class, and like I just don't know how worth it is to like really dive in.
Yeah. We'll talk during neuropath. We'll spend like probably half our time on different spinal brain lesions, like the ones that are most high yield. And then we'll spend half our time on kind of other neuro conditions. So I would not go too in the weeds on them. I would say it's probably not worth your study time.
Okay, thank you.
Yeah. Great. Any other questions here before I let y'all go?
I have a quick question.
Yes, Rita, of course.
So biochemistry is kind of my difficult point. And so say I write it down and then we do the next half of the test. Am I going to lose those notes and then have to remember it during that lunchtime, or how does that go?
Oh, yeah. So you will lose those notes. So whatever you write down on your first exam, that's that exam booklet for the first exam. You will turn that in and you will never get that exam booklet back again. After lunch, you'll get a new exam booklet for part 2.
Okay, so then we have to write it down again.
If you are writing down, yeah, if that's your part of your process is you're going to write out some of these pathways or components as part of your test taking strategy, you would need to write that out again if that's a part of your test taking strategy or test taking process.
Okay, great. And so then my last question is, I thought we didn't have to do diagnosis.
You won't be doing diagnosis, but you will be asked about pathology and you will be asked about pathologic conditions and high yield lesions or high yield pathophysio components. So we won't necessarily be diagnosing someone. They will ask potentially about clinical symptoms. They could ask about epidemiology, and they definitely can ask about pathophys. So that can be seen on this test, but you're not going to be asked what are the diagnostic criteria for rheumatic fever or for PCOS. But you could be asked, what is the hormonal association with PCOS? Because that's, even though it is diagnostic, it's also a part of its pathophysiology. Does that help?
Yes, thank you.
Yeah. And then the genetic SNPs, it's, even though it feels very diagnostic, it's what disease is associated with this genetic mutation. It's like, well, that feels like a diagnosis, but they hide that under the guise of genetics or pathophysiology.
On the 2 parts of the test, is that organized in any way, or is it all just 2 blocks of random questions?
2 blocks of random fun. Yep. Yeah. It is split up into 100 questions and 100 questions. So it's broken down into 50 case or 25 cases and 25 cases, but there's no rhyme or reason for type of case or type of question. So you can see everything on all of them.
Got it.
And they don't necessarily have equitable division of question type or case type on the test. So I found on my first test, I had no renal and no really like uro or repro. And on the second half, it felt like it was all of that because the first half didn't have any. So, but it ended up working out within the percentages. It just felt more heavy on that in the second half because I had none in the first. But that changes from year to year.
Got it.
Yeah. So there is no way to help focus like, oh, this part I'm going to focus on biochem for part 1 and part 2 I'll focus, that they don't organize it that way. Great. Any last minute questions before I close this down for today?
One last question for me.
Yes, of course, Raina.
So at the beginning, I kind of got overwhelmed because you had this huge microbiology thing, and I filled it all out. And then I was kind of looking and it said that there was going to be a lot of microbiology. What are your thoughts?
On micro? Yeah.
Yeah, and I filled it all out and I just got really overwhelmed.
It's a lot. I'm impressed you did all that. That's great study. That's fantastic. There will be more micro than immunology on the microimmuno section. So microimmuno is 20% of the exam. So a good chunk of your test will be micro and immunology. It tends to be more micro heavy, and it tends to be more focused on bacteria and viruses than fungi, protozoa, etc. So my strategy I recommend for people is focusing on bacteria and virus first, and really only leaving the most high yield fungi or protozoa or parasites. And if at this point, I would probably even say, don't even focus on those. Just focus on bacteria and viruses. What I mean, focus on bacteria and viruses, know the common cause or etiology of a condition if it's microbial in nature. So impetigo, Staph aureus, those type of pieces. We'll go through those next week. So we will talk about them specifically, like pneumonias, meningitis, those pieces where they change depending on population type. And then ideally for bacteria, know 1 or 2 additional factors about the microbes that are associated with that disease. So in regards to Staph aureus, if you knew it was also gram positive and it was also catalase positive and you understood what that meant, great. or if you know it was gram-positive and cocci in shape, great. But I tend to recommend know a bacteria for the disease, and then know 2 things about that bacteria that make it unique. And that is where I would stick to for those bacteria questions. Viruses, same, know the main viral etiology of the conditions kind of on that list of NPLEX. If they have a viral etiology, it's more common. And then know the DNA, RNA, double-stranded or single-stranded for that virus. That's probably your most high yield piece at this point for viruses that you'll be asked about. We'll look at that next week in class.
Thank you.
Yeah, good question. Awesome. Well, it's 12:01. Thank you for being here today. I will post these lectures either within this hour or within like the 2 o'clock hour. And then tomorrow we will meet back in person at 8 a.m. normal time, normal jam. Thanks.
Thank you.
Thank you.